And we're actually extremely pleased to welcome today Stephen Liu, Benjamin Besse, and Alexandra Leary to present some, first, the, let's say, global vision on non-small cell lung cancer. A second, a more precise approach on what our lead product, Tedopi, can bring to this disease. And eventually, Alexandra will present most recent data on ovarian cancer, which is a new indication for us, that has been presented at ASCO very recently. Alexandra made an oral presentation at ASCO in Chicago a few days ago. So in few words, I would like to make a brief introduction on the company. This is a company I joined less than a year ago. And I'm absolutely convinced that this is a future great biotech company in France and in Europe, because we have incredible products. We have this product that we will go into details today, which is Tedopi. It's an immunotherapy, which is very promising, with already more than 700 patients that have been treated. And we expect to have a Phase III readout early 28. So it's very close now. And we have a second compound, which is lusvertikimab, which has delivered a positive readout in inflammatory bowel disease last year, in particular in ulcerative colitis. We're not going to elaborate on lusvertikimab today. This is not the purpose of this webinar. So just if we can share, Thibaut, the first slide, to give a brief overview of the company's pipeline. Of course, a forward-looking statement, as we're a listed company. This is compulsory. And then on the next slide, you will have an overview of our clinical assets, which are twofold. We have proprietary assets, which are lusvertikimab and Tedopi, and some partnered clinical assets with Boehringer Ingelheim and Veloxis, which is part of Asahi, the Japanese company. As you can see, for lusvertikimab, we already have delivered Phase II data in ulcerative colitis last year. We're now preparing a subcutaneous formulation that is most suited to the actual markets, the actual needs of the patients. And we are planning to launch rapidly now a second indication, which is some kind of subset of ulcerative colitis. The second indication is called chronic pouchitis. It has the potential to read out in 27. And we could imagine to launch a product in few years. For Tedopi, the beauty of our pipeline is that we have this Phase III ongoing, ARTEMIA, on which Benjamin will spend some time. And we also have a series of investigator-sponsored studies, in particular the one that has been announced and described by Alexandra at ASCO on ovarian cancer. But we'll have additional readouts in the second part of this year, H2 26, in non-small cell lung cancer in a larger population than ARTEMIA. And we also have long-term readouts of pancreatic cancer trial. So now we'll leave the floor to Stephen, who will give a vision on the pathology itself. And thank you so much to all of you, Stephen, Benjamin, and Alexandra, for taking the time to make this presentations that will hopefully give more insights to our participants. Thank you so much.
Well, thanks for having me. My name is Dr. Stephen Liu. I'm a thoracic medical oncologist at Georgetown University in Washington, D.C., where I'm the chief of hematology and oncology and the head of developmental therapeutics. And what I'd love to do to set the scene for some of my colleagues is describe the current state of the treatment of lung cancer and how far we've come, and really outline the unmet need. So if we look at our slides here, I believe I start with some disclosures. And, in the past 13 years that I've been at Georgetown and treating lung cancer, we can go to the next slide. Past 13 years, we've clearly made a lot of advances in the treatment of lung cancer. We've really seen a wide reversal. Lung cancer is a fairly common disease. These are U.S. numbers, where you can see it's the second most common cancer in men behind prostate, the second most common cancer in women behind breast. And if we click ahead, while it's the second most common, it is by far the most lethal. We click ahead to the next. It's by far the most lethal cancer. You can see the number one cause of cancer-related death for both men and for women. As you can see, it represents more deaths each year in the U.S. than prostate cancer or breast cancer or liver cancer or ovarian cancer combined. So it remains a huge unmet need and one of the most lethal cancers that we have. If we go to the next slide. Here we can see that in red on the left, we see the mortality for men and for women. Lung cancer is on the way down, fortunately, but still far outpacing any other cancer in the US. On the right there, you can see the initial stage of diagnosis. These are 2026 numbers. And what we see is part of the reason is we detect lung cancer at a fairly late stage, with the most common stage in the US being stage 4. Outcomes for stage 4, as one could imagine, are clearly worse. And if we look at our survival numbers, while they are better for localized and regional, they're still fairly poor across the board. Our 5-year survival rate of 28% pales in comparison to other cancer types. We look at breast cancer, 5-year survival rate's 92%, prostate cancer, 98%. Colon cancer, a very important cancer, has a 5-year survival rate of 65%, and at lung, we are at 28%. So while we are proud of all the advances that we've made, and while these are the best survival numbers we've ever seen, we still have a long way to go, and this still remains an incredibly lethal disease. If we go to the next slide. We've seen advances in the treatment of lung cancer really in the form of two different paradigms. One is the development of targeted therapies, following on the heels of the Human Genome Project and the understanding that certain genomic subsets of lung cancer can respond very well to novel targeted therapies. Here we see real advances in medicinal chemistry, and if we can identify certain subsets of lung cancer, we can deliver very effective targeted therapies. Things like EGFR, ALK, ROS1. When we find these alterations, when we deliver a targeted therapy, we expect responses for the vast majority of patients. While there are some exceptions, for most of these patients, we will see resistance in 1 to 2 years, which remains a real challenge in the clinics. But importantly, this is only a subset of patients. For most patients with advanced lung cancer, immunotherapy will be the backbone of our treatment. This is appropriate for most cancers and across both histologies. For some, we use monotherapy, PD-L1 or PD-1 inhibitors alone, and for most, we use combinations, either dual immunotherapy or combinations with chemotherapy. But immunotherapy really is the mainstay for most patients with advanced lung cancer. And it's made a huge difference. And not only has it changed how we treat lung cancer, it's changed how we think of lung cancer and really raised the bar in terms of what is possible and what our expectations are. On the next slide, we get into some of the details regarding immunotherapy. And KEYNOTE-24, I think, is one of the more important studies that changed our standard of care. This was a study presented at ESMO in 2016. In this trial, we compared a single immunotherapy agent, a drug called pembrolizumab, a checkpoint inhibitor, an antibody targeting the PD-1 protein. This immunotherapy agent alone compared to standard chemotherapy that had been our approach for the treatment of lung cancer for decades. Chemotherapy that prolongs survival, improves quality of life. Here we compared that to immunotherapy. And what we saw from that Phase III trial was immunotherapy was better in every conceivable way. Better in terms of response, progression-free survival, the durability of control, and overall survival. Importantly, it was also better in terms of toxicity. Immunotherapy became our standard of care based on this trial for patients whose lung cancer had high expression of a protein called PD-L1. That's about a third of patients. These are updated data looking at 5-year survival rates. Let's pause there and acknowledge the importance of that sentence, 5-year survival rates. Decades ago, we wouldn't even conceive of anyone surviving 5 years with a stage 4 lung cancer diagnosis. And here, what you can see is almost a third of patients alive at 5 years. This study was presented in 2016, and so we have even longer follow-up. On the next slide, we see 8-year survival data. At 8 years, we have almost a quarter of patients alive at 8 years. So here we see 24% of patients at 8 years. Notice that there's not a whole lot of difference between the 6-year survival rate and the 8-year survival rate. But I think a better mark is how many of those patients are not have not seen any evidence of progression. If we look at the next slide, while 24% of patients are alive at 8 years, we see 13% of patients are progression-free. 13% of patients. So in this study, patients received 2 years of immunotherapy and then are observed. And for 13% of patients enrolled in this trial, 6 years with no therapy, they have still not seen their cancer come back. Now, the word cure is a tricky word. It's an emotional word and a hard word to define. But are these patients cured? And I believe that some, if not all, of these patients are cured. And so we are able to cure some patients. But notice it is the small minority of patients, only 13% of patients. But it is possible. And now, so to speak, we've bitten the apple. Now, this is really the bar. This is what we're capable of. And what we want to do is deliver this to more patients, more than 13%. But also remember that this study was done in patients with high PD-L1 expression, which is about a third of our patients. What about the other 2/3? What about most people with advanced lung cancer? And again, this is only a small subset. If we look at the next slide, we see long-term follow-up from a regimen called Keynote 189, which was immunotherapy with chemotherapy. This regimen looked at standard, state-of-the-art chemotherapy, platinum plus pemetrexed, either with placebo or pembrolizumab. And what we saw was adding immunotherapy to chemotherapy improved progression-free survival and, importantly, improved overall survival. And as we saw with the prior trial, it led to a chance of long-term survival. Here, the 5-year survival rate, almost 20%, almost 1 in 5 alive. And this includes all PD-L1 strata, so really an entire all-comer population within a non-squamous histology. On the next slide, how many of those patients are progression-free at 5 years? It's the minority. It's about 7 to 8%. And breaking it down by PD-L1 expression, for PD-L1 high, 13% of patients progression-free at 5 years. For PD-L1 low, about 7% of patients progression-free. For PD-L1 negative, only 2%. It's a small minority of patients, but what we've seen with immunotherapy strategies and recapitulated across many different immunotherapy agents is long-term survival and long-term progression-free survival is possible. It is not most patients, it is certainly not enough patients, but it is some patients. Now, this is what we're chasing for all patients. All of our patients deserve this opportunity, and so immunotherapy remains our standard of care for every non-small cell lung cancer that does not have an actionable target, which is most lung cancer. All patients deserve that opportunity. Most will not achieve this result. And what our job is, what our vision, our goal, our hope, our dream is to deliver this to all patients. And how do we get there? If we look at the next slide, the overall summary here is that most patients do not respond to immunotherapy. A small subset do achieve durable response, but another subset achieve a transient response. And this, I think, intellectually is very intriguing. On the next slide, we see follow-up from that Keynote 189 study. And in dark green, you'll see the course of therapy, 2 years with pembrolizumab. If we click ahead to the next slide, in dark green here, we see the 2-year course of pembrolizumab. And what we see with the black dots, those represent progression events. While some patients have progression of their cancer early, some patients do have it late. Some don't have it at all. That's certainly what we hope for, but some do progress late. These are instances where we feel we're very close to getting that immune response, where we were able to engage our immune system, but not in a durable way. This is really what we want to achieve, more durable immune responses. And for patients whose tumors initially responded but lose response, we really want to recapture that response. Because what is our alternative here? For patients who initially receive chemotherapy and immunotherapy and then progress, the current standard of care is docetaxel in the US, docetaxel with ramucirumab. This is a tough regimen and the outcomes are poor, but it is a regimen that we have not been able to surpass. When we look at the outcomes with standard docetaxel and ramucirumab, the response rate is 23%, only a fraction. And with docetaxel alone, which is the global standard, only 14% of patients will respond. 14%. The progression-free survival with docetaxel alone was 3 months, which is really the first scan. Any benefit is fairly transient, and the survival remains between 9 to 10 months. Outcomes with docetaxel are very poor, and the safety leaves a lot to be desired. Patients do not feel well with this treatment. Fatigue is seen in most patients, with 14% having grade 3 or very severe fatigue. Patients do not feel well. Neutropenia, or low white blood cell count, seen in 55% of patients. Most of those, almost all, are grade 3 febrile neutropenia, dangerous complications seen in 16% of patients. And about a third of patients require some dose hold or reduction or adjustment in some way. This is a regimen that we do not like prescribing. It has a ceiling on how well it can do. Patients do not feel well with treatment. We need something better to offer to patients, but we haven't seen anything better for quite some time. We're long overdue for an advance. And while we appreciate the advances with immunotherapy, we're ready for something more. And so to wrap up on the next slide, where we see things currently with non-small cell lung cancer without a driver alteration, immunotherapies are standard of care, clearly superior to chemotherapy alone, and importantly, offers the potential for long-term survival. We celebrate this advance. We offer hope to our patients, but for most, the likelihood of long-term survival is quite low. So we can celebrate the advance while still acknowledging the unmet need is pressing and looms large. Progress has stalled. While immunotherapy is state-of-the-art, the Keynote-24 was FDA approved in 2016. Chemotherapy with immunotherapy in 2018. Almost a decade since some of these have been approved, and we still haven't improved upon that. The pursuit of transformative change really lies in finding durable survival. And what we do not want, what we do not need, are incremental benefits with cytotoxic molecules. What we want for patients are more immune-mediated, anti-tumor responses that have meaningful, durable survival and progression-free survival. This is really what we're chasing, and our hope is that this program will get us closer there. And so with that, I'm happy to turn things over to the next part.
Many thanks, Stephen. Now we're passed to Benjamin, Institut Gustave Roussy.
Sure. I'm happy to be there. I'm Benjamin Besse. I'm a medical oncologist, mostly thoracic oncologist, and head of clinical research at Gustave Roussy. And now we will talk about therapeutic vaccination and Tedopi in patients with non-small cell lung cancer. So there are two main kinds of vaccines, those that are ready to use or off the shelf, which are vaccine already prepared, that can be based on protein, on mRNA, and Tedopi is one of them, with a mixture of epitopes, and it's dedicated to patients that have an HLA-A2 positive phenotype. Other vaccines are personalized. Usually they are RNA vaccines. They are quite costly and difficult to make, with a turnover time that is usually between 6 and 7 weeks, because you need a biopsy or a surgical specimens of the tumor, and then you generate the vaccine, the personalized vaccines. So we will talk about Tedopi that is a ready-to-use one, as I said. So next slide. What is Tedopi? It's a combination of neoepitopes. It's 9 optimized neoepitopes that will cover roughly 100% of non-small cell lung cancer. So you don't have to adapt the vaccine to the expression of the epitope. It's ready to use to any kind of non-small cell lung cancer, squamous or non-squamous. There is a binding to HLA-A2, so we have to restrict the population to HLA-A2 positive patients, which represent 45% of our patients. So it's a ready-to-use emulation that is given subcut each 3 weeks. Next slide. The first phase II study was done in 64 patients with non-small cell lung cancer, and all these patients were chemo pretreated. And the overall survival of the vaccine is quite impressive in this pretreated population, 17.3 months. It's obviously restricted to HLA-A2 positive patients, but interestingly, in this sub-analysis, OSE look at the benefit of the vaccine based on the epitope response. So you measure in the blood the immune response to each of the 9 epitopes. And the more immune response you have against epitopes, the better is the survival, which is a strong indication that the activation of the immune system, the specific activation of the immune system by Tedopi, leads to a disease control and a benefit of overall survival. The second point is, you remember all these curves on immunotherapy in first line that Stephen showed. All these curves, as he highlighted, a plateau. So the shape of the curve is really that you have long responders in these patients. And interestingly, even in the patients with a lower epitope response, you can see the shape of the curve with long responders. So next slide. These drive to the development of the first phase III trial in patients with non-small cell lung cancer pretreated with chemo and immunotherapy. It's the phase III ATALANT-1 trial. Next slide. So the trial was done in second line, as Stephen presented. Now, when you diagnose a non-small cell lung cancer, you have to look at the targets that make the patient eligible for targeted therapy. That will be roughly 15% of the Caucasian population, and all the other ones will receive immunotherapy or chemoimmunotherapy. But at the time of progression, there is only one standard of care, that is docetaxel, a standard single-agent immunotherapy, quite toxic, I have to say. Next slide. There have been a lot of attempts to beat docetaxel as a standard of care. And a few years ago, we had a lot of expectation with two types of strategy: VEGFR inhibitors, and this is example of two phase III studies that have combined an immunotherapy plus a VEGFR inhibitor and randomized against docetaxel. The two trials are negative for overall survival, but there are 4 randomized trials with 4 different combinations of an IO plus a VEGFR inhibitors, and all these studies are negative. The other class of drug that is really under the spotlight is the ADC, the antibody drug chemotherapy. So these are agent that were given single agent and combined head-to-head with docetaxel, second line, again, post-chemo immunotherapy. And none of these drugs made it, even if some of the trial were statistically significant. The clinical benefit was not enough to have an FDA or EMEA approval. So that now, standard of care, even with all this development of new drugs, standard of care is still docetaxel for the second line. Next slide. So in the ATALANT trial, Tedopi was randomized 2-to-1 to docetaxel, the standard of care, in exactly this population, which we just described, so patients with advanced or metastatic non-small cell lung cancer, with an HLA-A2 positive test. EGFR mutation and ALK fusions were excluded. Any PD-L1 stated could be enrolled. All the patients had to progress under chemo, immunotherapy, given sequentially, or both combined, but the last line had to be the immune checkpoint blockers. So the patients were randomized between Tedopi and docetaxel, and the primary endpoint was overall survival. Next slide. So the trial aimed to recruit almost 320 patients, but because of the COVID pandemic and the new vaccine for COVID, the trial was stopped after inclusion of 219 patients. These patients had first or secondary resistance to immunotherapy, meaning that they received less or more than 24 weeks of immunotherapy. The primary population that we defined was those patients with secondary resistance, so all these patients that receive at least 42 weeks, 6 months roughly, of immunotherapy, and that represent 118 patients. Next slide. So in this primary population, if we compare to the intent to treat population, the patient's characteristics are overall quite the same. The percentage of squamous, in particular the TNM stage at study entry, or the rate of brain or liver metastasis, it was roughly similar. Next slide. So in the primary population, these patients with secondary resistance to immunotherapy, you can see a significant and clinically significant improvement of the overall survival, 7.5 months in the control arm, what you expect, versus 11.1 months in the Tedopi arm, with an hazard ratio of 0.59. Next slide. If you look at the subgroup where you could see a benefit, the only subgroup where there is zero benefit, apart from the stage 3, but as you can see, it's a very, very small population, so difficult to have any conclusion. It's the patients with brain metastasis. So in the current trial, these patients are excluded. Next slide. If we look at the time to worsening of the performance status, it was much better in the Tedopi arm compared to docetaxel arm, and this related to both the efficacy and the toxicity. Next slide. If we look at the toxicity of the docetaxel or Tedopi, it's striking to see that you have 3 times less severe grade 3 to 5 adverse events with the vaccines compared to docetaxel. So in terms of toxicity profile, the vaccine is much better in a lot of parameters, hematologic toxicity, but also very important parameters such as alopecia, docetaxel-induced alopecia, where a vaccine doesn't. And this is also why the quality of life was better in the Tedopi arm compared to the chemotherapy, both because it's more effective and because you have less toxicity. Next slide. So as I said, this trial was stopped because of the pandemic, and the subgroup analysis of the secondary responder shows strong benefit, but it was not enough to put the drug on the market. It was enough to have a very strong hypothesis to build a new phase 3 trial in a much more precise population, the patients with non-small cell lung cancer, pretreated with chemoimmunotherapy, HLA-A2 positive, and with secondary resistance to checkpoint inhibitors. Next slide. So this is the main characteristic of the trial, the population, HLA-A2 metastatic non-small cell lung cancer with secondary resistance to immune checkpoint inhibitor, defined by at least 24 weeks of immunotherapy. Docetaxel was accepted by FDA as a standard of care for the comparator arm. The primary endpoint is overall survival. This is really meaningful and convincing for such trial. Among the main secondary endpoint, the patient-reported outcome and quality of life was accepted by FDA. There is a central HLA-A2 testing with a companion diagnosis test. And this leads to, next slide, this design. So in the ARTEMIA study, an ongoing phase 3 study, Tedopi is again compared to docetaxel, but in this well-defined population, HLA-A2 positive, squamous and non-squamous non-small cell lung cancer, pretreated with chemo and immunotherapy and with secondary resistance, so at least 24 weeks of immune checkpoint inhibitor. And as I said, because of the subgroup analysis, the brain mets were excluded. Primary endpoint is overall survival, and there is a stratification on histology, squamous and non-squamous, and performance status. And the trial is now ongoing. And while I will stop here and give the word to Alex, that has presented the results in the ovarian cancer.
Many thanks, Benjamin. Up to you, Alexandra.
Thank you. So I have a first slide. So I'm Alexandra Leary. I'm a medical
actuellement la
oncologist.
conférence.
I'm a medical oncologist at Gustave Roussy and deputy head of the Department of Medical Oncology. And I'm also the president of the National Intergroup for Clinical Trials in Gyn-Onc. Next slide. So before I jump into the trial, because we've talked only about lung cancer so far, I'm just going to give like a 30-second introduction on ovarian cancer. Ovarian cancer is not as frequent as lung cancer, but it's deathly as well. 80% are diagnosed at an advanced stage where we do chemo and surgery, and for years, the 5-year survival without disease in ovarian cancer was less than 2%. So when we're talking about cure rates, even in upfront surgically resected ovarian cancer, our cure rate was 2%. So absolutely horrible prognosis. This has clearly evolved with better surgery and the advent of targeted therapies such as PARP inhibitors and bevacizumab. And actually today, by putting chemo, surgery, and using these maintenance options, we've dramatically improved survival, with now 5-year disease-free survival close to 40%, which is great. And that's excellent news. For years, when they relapsed, we would do platinum again and platinum again and platinum again. That was our favorite chemo. It worked every time you put it in again. Unfortunately, by doing much better in first line, those who are not cured in first line and progress under the pressure of something like a PARP inhibitor or after bevacizumab, suddenly when we were putting a platinum again, it wasn't working as well. Okay. And we have no maintenance strategies for these patients. So with that in mind, I'm going to present the result of this randomized international academically sponsored trial testing the neoepitope-based vaccine OSE 2101 that you've just heard about, plus or minus an anti-PD-1 pembrolizumab versus best supportive care as maintenance in these platinum-sensitive relapse patients with ovarian cancer. Next slide. So again, why we did this study? Well, actually, platinum-sensitive ovarian cancer used to be easy to treat, and today it's an area of unmet medical need, okay? We have no approved maintenance strategies for these patients who relapse, who we treat again with 6 cycle of platinum, who previously received bevacizumab and previously received a PARP inhibitor. No more maintenance. We just have to do watch and wait. The other thing is there hasn't been a single positive randomized trial in this setting for platinum-sensitive relapsed ovarian cancer in years, over 5 years. And this is despite huge Phase III trials testing immune checkpoint inhibitors as maintenance on their own, with bevacizumab, with PARP inhibitors, all negative. And as I said, these patients who progress post-PARP have poor response when you reintroduce the platinum. And even if you manage to get some platinum in, we stop it usually after 6 cycles, they relapse really shortly thereafter. Next slide. So I'm not going to give again the background on this vaccine, which has been very well introduced. It's relevant in ovarian cancer because 80 to 90% of ovarian
Rejoins
tumors
actuellement la conférence.
express one or more of these tumor-associated antigens. And it's important, they're not just tumor-associated antigens. These have been modified to increase their binding capacity to the T-cell receptor and or to HLA-A2. And given the positive results in lung cancer with acquired immune checkpoint resistance, and the fact that so far immune checkpoints alone or with PARP or with BEV weren't being efficacious in ovarian cancer, we thought we would use this vaccine to turn what is frequently described as an immune cold tumor into an immune hot, okay? And that was the aim of the study, was to evaluate the benefit of combining this vaccine with an anti-PD-1 as maintenance after platinum in this very difficult to treat patient population. Next slide. So the design was rather simple. Basically, any patient who is relapsing and eligible for platinum doublet, received a platinum doublet 4 to 6 cycles, and had at least their disease controlled, so stable disease, partial response, or complete response, who had previously had a PARP inhibitor, previously had BEV, or had a contraindication. Therefore, there's no maintenance options for these patients. Then, if they were HLA-2 positive, they were randomized to the standard of care, which unfortunately is watch and wait, or best supportive care, versus the vaccine on its own versus a combination. And the randomization was 1 to 1 to 2. Next slide. Without going too much into the statistics, there was three arms, but we designed the trial to have, with a sample size of 185 patients, to have the power to detect an important difference between the combination of the vaccine and pembro versus best supportive care. Okay. Next slide. The patient characteristics, I probably won't go into too much for those of you who are not particularly ovarian cancer specialists, but they're as we expect. They were very pretreated with PARP, pretreated with BEV. Roughly half of them had stable disease, half of them were in complete or partial response to the chemo when they were randomized. Next slide. And these are the primary results, okay? The combination of vaccine with pembrolizumab as maintenance after platinum significantly decreased the risk of progression by 50% compared to standard of care, with a hazard ratio of 0.53 and a highly significant p-value. So the trial is positive. We achieved the result we were hoping to get. So that's for the primary endpoint. Next slide. So although we weren't powered to do this, we couldn't help but look. So when you compare the three arms between each other, there's an incremental benefit for the combination compared to the vaccine on its own, and there's an incremental benefit for the vaccine on its own versus best supportive care, although this did not reach statistical significance. Next slide. The safety was actually interesting. So the vaccine had been previously tested and very well tolerated, as Benjamin said. We did have more immune-related toxicities in the combination. And we know an anti-PD-1 alone, vaccine alone, and we were seeing more immune-related reactions with the combination than we expected, which we actually, while the trial was going on, considered a good sign that we were actually stimulating the immune system. The most common grade 1 or 2 were local site injections, okay? And having seen some of Benjamin's patients, who are men and women, at least in my women-only population, it seems they get more local site reactions. Grade 1 or 2, but, you know, they're there. The intriguing one was the cytokine release syndrome. They were very rare with the vaccine alone in previous studies. It was very rare in our vaccine alone arm here, with 9%, mainly grade 1, 2. But we were seeing more, mainly grade 1, 2, cytokine release syndromes with a combination of vaccine and an immune checkpoint. Okay. Again, at least proving that we were achieving what we wanted to achieve. But therefore, halfway through the study, we said, okay, we're going to put just very simple primary prophylaxis with basically paracetamol and non-steroidal anti-inflammatories. And with that, we significantly decreased the risk of grade 1, 2 CRS. Next slide. When you look at the subgroups, the benefit was seen across the subgroups. If you look at HRD negative all the way at the bottom, it suggests no benefit. There's 13 patients. I think you should, and in a multivariate, it was negative and there was no interaction. On multivariate analysis, the only group that appeared to have especially great benefit were the patients who were in partial or complete response to chemotherapy, where there the hazard ratio was 0.31, which is really quite remarkable. Next slide. So in conclusions, we present the first trial providing a proof of concept for the benefit of a vaccine approach in ovarian cancer. This is also the first positive randomized trial in the platinum-sensitive setting ovarian cancer in years. And the combination of the vaccine with pembro as maintenance after platinum significantly reduced the risk of progression by 50% and by almost 70% in those in CRPR. There was a little bit more toxicity, but it was mainly grade 1, 2, and quite manageable once we introduced really simple prophylaxis. And this study, its control arm confirmed that these patients who relapse, they get another platinum. Their prognosis is terrible if they have prior PARP exposure. Once you stop the platinum, their PFS is less than 3 months. And we really feel this supports a Phase III confirmatory registration trial. And then last slide. Do I have any more? Next one. And this was sort of the lay summary. It's just a reminder that women with ovarian cancer who receive platinum-based chemotherapy when their cancer comes back after prior Bev and PARP have a poor prognosis. Okay, and that's on the, let's say, the flip coin of the fact that we're doing such great treatment in first line. Fortunately, if that doesn't cure them when they relapse, they're harder to treat. The TEDOVA trial showed that giving a combination of an anti-tumor vaccine with immunotherapy as maintenance after platinum significantly delayed cancer progression. As I said, this is the first positive randomized trial in years for this patient population and the first proof of concept. So we're really excited about this data, and we can't wait to go to the next step. Thank you very much for your attention.
Many thanks, Alexandra. So now we are moving to questions. First, I would like to thank, in particular, Valérie Gabard, our head of MSL, and Thomas Gidouin, our CFO and Deputy CEO, who have prepared this webinar. So I think that we have a first question coming from Aaron. Aaron's asked the question. So I'm not so familiar with that, so maybe I will read the question. When we look at the population of interest for Tedopi's evaluation in non-small cell lung cancer, don't you think that we address Tedopi at a very restrictive population, with the double restriction of HLA-A2 phenotype and secondary resistant to checkpoint inhibitors? Benjamin, maybe can you answer this question?
Sure. So since more than 20 years now, we are heavily used in non-small cell lung cancer to treat subgroup of patients that are started with EGFR inhibitors and then ALK and then ROS1, and we have now 10 targets that we have to screen in all the patients. And some of these targets, like BRAF inhibitors, BRAF mutation, is 1% of our population, and that's enough to develop a randomized trial and to approve drug on the market. So in lung, we are very, very used to screen for markers and treat subgroups. So now we think that this population will represent roughly 15% of the patients that progressed after chemoimmunotherapy, which is a huge number for you. So I don't see that as a limitation.
Thank you for that. So, sorry, Stephen, sorry.
Yeah, echo Benjamin's thoughts here. We're used to dealing with small populations. I'll mention in the US, HLA-A2 is 40 to 50% of patients. This is the most common allelic subtype as well. And you have to start with where the drug is going to perform best and follow the science. And I think that's the starting point. But if this really confirms our suspicions that this is an effective treatment, I think that it opens the door to further modifications, which can sort of expand its use in different indications. But the HLA-A2 is the most common subtype.
Thank you so much for that. I think that Aaron is connected. Would you like to ask oral questions? If we can do that technically, I'm not familiar with that. So, not sure it works like this. Thibault.
Hi, can you hear me?
Oh yeah, excellent.
Fantastic. Thank you for the event today, and thanks for taking my question. Curious to learn if the ARTEMIA trial results are positive. How quickly do the KOLs think that Tedopi could fit into the second-line treatment pathway? And what, if any, physician education or diagnostic infrastructure would be needed?
I can start with the US answer very quickly. Again, the endpoint here is overall survival, and we are eager for something to replace docetaxel. As Benjamin outlined, there have been many large Phase III trials that have simply failed to improve outcomes. And so if we do demonstrate an OS advantage, I think that there would be a very enormous appetite to move this forward fairly quickly, first on guidelines, and then certainly through the FDA process. HLA subtyping is done pretty routinely as part of most of our commercially available next generation sequencing panels. So in the US, we often know that information up front, and we wouldn't have to have big changes in terms of our current testing practices, at least for most institutions. Certainly the timelines would be a little different in Europe, I imagine.
Regarding Europe, HLA-A2 is a very simple blood test, so I don't see any issue to have HLA-A2 as a reflect testing. It's not, I mean, it's something that is intrinsic to the patient, so it's not something that appears at resistance, for example. So you can screen upfront when the patient is diagnosed or start his first-line chemo-IO, and when you have a doubt of progression. So the testing will be something very easy. And as Stephen said, we all want to give something else than docetaxel. So I think the uptake of such vaccine would be very massive.
Thank you for that. Aaron, do you have additional questions?
Yes, thank you very much. One more question from me, perhaps that's for Alexandra. Looking at the ovarian cancer program, the data was really encouraging, so thanks for presenting that. Please, could we hear your thoughts on what a credible subsequent study might look like in terms of design and how it would build on from the prior trial?
So that's a very good question. It is a matter of discussion because we just got these results. When we designed this trial, the acceptable standard of care, the only available standard of care in this setting was best supportive care. Today, it's becoming increasingly unacceptable not to give any maintenance, okay? Even though we don't have reimbursements, which is a bit of a problem. So one idea would be to say, well, you know, there is data to support putting an anti-angiogenic in people who are previously exposed to bevacizumab. And would that be appropriate to add an anti-angiogenic component both in the control and the experimental arm? PARP inhibitors, we don't have any data, but we have a large phase 3 study that supports Bev after Bev or anti-angiogenic after angiogenic. So that would be one slight modification, which I think ethically and even just it makes sense.
Perfect. That's very helpful. Thank you very much. No more questions from me, but look forward to following the progress.
Thank you for that. So we have written questions. With the rapid emergence of other approaches like ADC, bispecifics, next-gen immunotherapy, where do you see Tedopi fitting into the treatment algorithm in the coming years? Do you view Tedopi primarily as a standalone therapy, or do you believe its greatest potential could be coming from combination strategies with checkpoint inhibitors, ADCs, other immunotherapies?
Well, I may start. When we designed ARTEMIA, a lot were thinking that ADC would be the standard of care, and it's not, on the phase 3 are negative. But other strategy also are not there. You're talking about bispecific. Genmab had a randomized study with Gen 1046, which is a PD-L1 4-1BB bispecific antibody, and they stopped the phase 3 study. It seems to be for not based on data, if I understood correctly, but they stopped the program. Latify was also a very promising study with ceralasertib, an ATR inhibitor combined with IO in second line compared to docetaxel, and the trial is negative. So the signal that we have from Atalant-1 is quite strong in a situation, in fact, where all the phase 3 so far fail. And I don't see, and maybe Stephen, you have other idea, but I don't see an asset that have, let's say, much more activity than the one that I just alluded to. So no, I don't see in the upcoming years the standard of care changing.
Yeah, I think that ADCs have shown a lot of promise really across tumor types, but at their heart, they are still cytotoxic molecules. While they can offer responses, they still share a lot of toxicities that we see with standard chemotherapy. So I think that will be a challenge to a lot of patients, particularly in the later line settings. I think that the difference with Tedopi is it really is an immune-based strategy. It can really make a big difference both in terms of quality of life and overall survival. These immune-based strategies offer, I think, the potential of much more durability than we might see with something like a cytotoxic agent. So ADCs, there are a lot. It's a crowded field. I suspect just by sheer number that one of these studies will be positive, but how much of a huge impact it'll make remains to be seen. I still think that there's an appetite for an immune-based strategy. In terms of monotherapy versus combinations, I think once you demonstrate that this offers a survival advantage as monotherapy, then you open the door to all sorts of combinations. And whether that's with bispecifics or with checkpoint inhibitors, could that further long-term survival odds? And could that really enhance those immune-mediated anti-tumor responses is something that I'm very optimistic for. Of course, first we have to demonstrate clear efficacy as a single agent before we explore combinations, but I see no reason why this wouldn't be able to be combined with other strategies.
And in terms of the ovarian cancer space, I sort of have the same answer. As I said in my introduction, people have tried to get a new standard of care in maintenance platinum sensitive, and they have not managed with immune checkpoint inhibitors on their own, or when they combine them with what we thought was the best, such as PARP inhibitors. It didn't add anything. So there's not so much out there. There are ADCs that are being explored everywhere. They're putting them in every indication, and a lot of them as maintenance. Let me tell you, an ADC as maintenance is not the same thing as an immune therapy as maintenance in terms of quality of life. Okay, and so for now, it'll be a while before we get even the opportunity of a positive study with an ADC in the platinum sensitive setting. And we do believe in combining it because we've already combined it with an anti-PD-1, and I think it might be worth thinking about combining it with an anti-VEGF. But I think it's nice to have a maintenance that doesn't have a cytotoxic in it.
Thank you for that. An additional question regarding TEDOVA. Did patients presenting CRS benefit better from the treatment from Tedopi than others?
It's an excellent question. No, we couldn't see that. And one of the reasons is that in the early part of the trial, we didn't expect CRS. And gyn-oncs haven't done a lot of CAR T cell therapies and TCR modified T cells. So when they saw CRSs, a lot of the investigators were a little shocked. And my concern was they were going to stop the treatment. That's why I introduced the prophylaxis. Okay. However, when we look at prophylaxis, people who came in sort of in the second phase and had prophylaxis, they seem to benefit more. Probably because those patients with CRS, who probably are good candidates, once we had the prophylaxis, they had very low grade CRS and they continued. Okay. So it's sort of a sweet balance between causing an immune reaction, but we don't want it to be scary enough that the investigator or the patient says, I don't want any more. And I think by introducing the prophylaxis, we helped with that.
Okay, very clear. Thank you for that. Another question regarding TEDOVA, sorry. You mentioned a poor prognosis, Alexandra, and what is in your, let's say globally, the current median overall survival, and what would be something that you would define as clinically relevant, assuming that you will get this data, I don't know, probably next year, later this year or next year. What would be the median overall survival improvement, let's say, clinically relevant for you?
So demonstrating improvements in overall survival in ovarian cancer in clinical trials has been challenging. Okay. Finally, we've arrived there for the first time in, I think, 3 decades, a couple of weeks ago, in the resistant setting. Okay. So the resistant setting is not where I am here. It's patients, they've become resistant to platinum. And here, the standard chemotherapy doesn't work. It's a little bit like the docetaxel, right? And so they don't have subsequent lines of therapy. And so if you have a PFS benefit in that setting, it is easier to translate into OS. And so finally, we have 2 positive clinical trials in the resistant setting that showed an OS benefit. But all of our studies in first line or subsequent sensitive setting, we haven't been able to translate PFS benefit and OS benefit because subsequent therapies still work. Now, again, this might change because here in the post-PARP setting, that might change. And so I don't like to talk about numerical benefit, but here we have a hazard ratio of 0.53 of PFS. If we get a hazard ratio that's positive, 0.7, 0.8, no one has ever achieved that in first-line or platinum-sensitive relapse. So any impact on OS, to me, would be a great success.
Very clear. More sophisticated question. I'm sorry, I will try to read that carefully. Give that Tedopi targets multiple specific antigens to turn cold tumors hot. Have the translational data from TEDOVA shown any evidence of epitope spreading to non-targeted antigens?
So the work is ongoing. So we have collected PBMCs. We're working with an academic lab to do all of this. And I haven't yet seen the data, so I'm not hiding anything from you. We'll be very interested to see. Some of the data that was already generated by Benjamin in the previous lung trials suggests that the epitope-specific response might be relevant. As to the rest, I don't yet know, but we have a very comprehensive translational program, as well as targets and the expression in the tumors themselves, the type of p53 mutations. We have two types. One is the loss, and the other one is overexpression. That's probably relevant. So there's a lot of work still to be done, and we look forward to presenting that in subsequent presentations and publications.
Fantastic. Thank you so much. I think we're coming to the end of this webinar. Thank you so much to the three of you for all your dedication to patients first, and to science globally. And we're very enthusiastic at OSE to move forward with Tedopi in, of course, in non-small cell lung cancer in the very short term, and assuming that we have financial capacities in ovarian cancer and maybe in other indications. So I thank you so much. And I thank all the participants to this webinar. It is recorded, so this will be available probably for the next few weeks on our website. And I wish you a great day, and see you and talk to you soon. Thank you again.